Good morning, everyone. So this is Tamalika Chakraborty from Guru Nanak Institute of Pharmaceutical Science and Technology. So I have participated in IMCCRT 2024. So I want to present my review paper on involvement of genes in neurodevelopmental disorder. So for that, this is my topic. And let me introduce first to what is neurodevelopmental disorders and why I have selected this topic. Because we know that in this era of genetics, where we have so many different kinds of new instruments for gene sequencing, millions and millions of DNAs are sequenced and genetic profilings are being done. But there are certain problems of the society which still remains unsolved, that is neurodevelopmental disorder. There are so many different atypical child in this spectrum who are suffering day to day with various kinds of difficulties. So that's why my topic today is to delve into whether this neurodevelopmental disorder is associated with genetics or it is having some different aspect, and what we can do in near future to overcome this kind of irreversible problem. So moving on to my next slide. Moving on to my next slide. So this is basically a brain. And this particular brain, which you can see that there are two different kinds of brain over here. And one is a typical brain and an atypical brain. So brain undergoes a remarkable process of restructuring. So as it adapts to the environment and become more efficient in processing information, there is some difference reported in the typical and atypical brain using post-mortem examination of tissue. So the age at which synaptic density peaked for a range of cortical areas was investigated by tracking changes in synaptic density at different ages. So there are few observations, and these observations are increase in brain volume, cortical thickness, callosal thickness. So in one paper in Hu et al., so it was a paper from Nature, have shown regional brain changes in a longitudinal data set from children finding expansion of cerebral white matter and shrinkage of parietal, temporal, and occipital gray matter. So it is no doubt a grave kind of disease and it needs an attention. So moving on to the next slide. So now my question to this is the third slide which I am talking about. So now my question is that, my question mark is that whether it is a gene or it is your environmental factor which is affecting it. Now, here why this question I just want to tell you, because those kids which were taken for the diagnosis of neurodevelopmental disorders, right? So these are, there are so many different views of doctors. Most of them do not target a gene, because when you have some genetic change, you have facial expression change. There is a change in the facial expression. But in this spectrum of disorder, like autistic spectrum of disorder or attention deficit hyperactive disorder, you do not have facial change. There are some behavioral issues, but you do not see any kind of facial change. So there comes the next question, that is it acquired during the lifetime? Is it due to placental oxygen deficiency during mother's pregnancy? So that is what question we will be delving into in this particular video. So moving on to my next slide. So next is that this is basically your neurodevelopmental disorder, which I was talking about. And what are the signs and symptoms of this neurodevelopmental disorder. So you get to see the signs and symptoms of neurodevelopmental disorder, which involves communication difficulties. So you have difficulties to communicate, how to communicate. So this child have speech delays. They do repetitive behaviors. Their behaviors are repetitive. They repeat the behavior one after the other, right? They have attention and focus issues, like they do not sit at a particular place and do not concentrate properly, and that's why they have learning delays. Challenges in social interaction, though I will have a controversial point over here, because always it's not that all atypical child have social problem, and that's why they are having a type of that they remain in one corner of a house, they do not interact. No, not like that. There are many child who even interacts, but communication difficulties, speech difficulties, speech delays, these are some of the very important part. And learning and sensory issues, hand flapping, walking on the top of the toe. So these are some of the atypical behavior which do not see in a in a child which is having a typical behavior. I will not say a normal and an abnormal, because these are basically the development of the arrangement of the neurons, their sensory input, which results in this kind of your output. So next is the risk genes. So moving on to, so when I have delved into this particular subject and have studied this particular subject, so I have came across some genes. These are basically, most of the papers are from the Nature Scientific Report. As I've seen that most of the papers, they are basically interested in, some of the scientists, they are basically interested in this genetic aspect of neurodevelopmental disorder. So you get to see like risk genes for ASD, which is your CHD gene. So CHD gene has been found in human, mice, and zebrafish. Zebrafish is also a very good model and robust organism in which you can do a lot of prospects in identification of neurodevelopmental disorder. So deformities of the face, skull, impairment association with the learning, memory, macrocephaly. So these are some of the complications which are seen when there is a mutation of CHD8 gene, though I will be discussing this gene in great details. Then in CNTNAP2, this is another gene again which was found in zebrafish. So there is a deficiency of GABAergic transmission, especially in the frontal region of the brain, and vulnerability of the drug-induced convulsions, abnormal neuronal development, diminished GABAergic neurons, unprovoked convulsions, restlessness, social impairments, and increase in repetitive behavioral tendency. So the repetitive behavioral tendency which I was talking about is basically from CNTNAP2. Moving on to the third gene, this is the most, walked upon gene, that is your SHANK. So there are three kinds of SHANK, SHANK1, SHANK2, SHANK3, which was found in human and zebrafish, and it has been found that the risk of developmental anomalies, an increase in atypical tail bending, weakened sense of social choice, an increase in swimming behavior, a general drop in fish locomotor activity. So locomotor activity is also impaired if SHANK is mutated. Next is your DYRK1A. So DYRK1A is again another gene which is associated with reduction in forebrain size, along with diminished midbrain activity, increased hypothermia-induced convulsion, as well as significant cognitive difficulties. So there are so many other genes I will be discussing in my next slide. So next is, again, get to see that this is my paper. Tamalika Chakraborty. We'll get it in International Journal of Pharmaceutical Science and Research. I have a lot of papers on neurodevelopmental disorder and also a lot of work on zebrafish regarding their behavioral antipsychotic activities of various drug and behavioral problems. So this is a table of neurodevelopmental disorder in which I have shown about some of the genes, right, and their location in the chromosome and different kind of mutation which was associated. So the first mutation is 3' UTR mutation. So this type of mutation is associated with increased risk of cancer. So there are so many different other kind of mutation like missense mutation, framesheet mutation, nonsense mutation, spliced sites. So different kind of mutation. Actually, mutation is diverse. You may have a deletion, you may have an addition, you may have a chunk deletion, you may have a chunk addition, you may have duplication. So there are so many different kind of mutation, and those mutation and associated gene which induce ASD. Try to differentiate that what is autism and what is ASD. Core autism is a bit different from autistic spectrum of disorder, because in autistic spectrum of disorder, which we call it as a regressive autism, where the brain regress, the activity regress, the improvement is very, very low. So lots and lots of genes are involved in it. And so it is very difficult for us to pinpoint that what gene to identify, what gene to, if we have any gene therapy, if we have any antisense therapy, so what gene to silence or what gene to replace. So that is very, very difficult. So this is again another neurodevelopmental disorder which I have talked about, and this is your ADHD, that is attention deficit hyperactive disorder. Again, this is from my paper, that is in International Journal of Pharmaceutical Science and Research, and I have given a bit of some of the examples of genes which are associated with the risk factor of ADHD. Like, for example, LPHN3. It has been tested in mice, zebrafish. So delay on cortical mutagenesis, altered neuronal structure and function, and altered timing of brain development. So altered timing of brain development, that is a very important point. So what is altered timing of brain development? Altered timing of brain development means that the development is occurring, but the time is different, the time is delayed. Then DRD4 and DRD5, which was seen in humans specifically, not in zebrafish, anger, irritability, attention-seeking property. So it is very, very, interesting that so many different kinds of genes are there which are actually controlling our behaviors. Then again, SLC6A3, that is again found in human, and orthologous gene has been found in zebrafish, which hovers close to tank's bottom, lowering immunoactivity neurons, behavioral problems, right? So decreased thickness of cortex, functional hyperactivity, and so on. And then we come to FHTIB, that is your found in mouse, zebrafish, and human, interface with dopaminergic signaling pathway of the fish, increase in impulsivity and aggression, increase in depression among young people, followed by rise in suicide. So it is really a very difficult thing. And now these are some of the points which I have highlighted on this gene in detail. So I will just go quickly through it because I don't have enough time. I think that chromodomain helicase DNA binding protein 8 has been identified as one of the strongest association, because these are transcriptional regulator that is expressed in nearly all cell types and implicated in multicellular process, including cell cycle, cell development, adhesion, neuronal development, etc. So even myelination and synaptogenesis. So it has been documented that CHD8 mutation exhibits a unique phenotype like large head size, typical face, broad forehead, flat nasal bridge, large ears, increased occipital circumference. So since if this kind of mutation is developing in a child and there is some facial expression, right, then it is maybe the genes which are responsible for autism. But there are many child in this spectrum, you will have absolutely normal texture, normal face, normal behavior, means normal, they look normal. If you take a picture of it, of a child of in this type of disorder, you will see that it's absolutely normal. So there comes a difficulty, that's whether it is genetic or it is environmental, though there are so many genes which are involved in it. So this is basically CNT NAP2 dependent molecular network in autism spectrum disorder revealed through an integrated multi-omic analysis. So I have given this particular paper, the screenshot of this paper, because multi-omics are also being used in identification of this spectrum. So people are trying to delve into this complex disorder. So in 2006, a recessive mutation in CTNP2 was identified in Amish family and is clearly syndromic form of ASD called cortical dysplasia focal epilepsy syndrome. So I don't know that how, whether this is not, I don't have an idea that if epilepsy is related to ASD, but in most of the child in ASD, I have not seen epileptic kind of behavior as per my understanding and my observation. So CDFE is rare neuronal migration disorder resulting in epileptic seizures, language regression, intellectual disability, hyperactive, and in nearly 2/3 of the patient ASD box 1. Subsequently, several laboratory provides covering evidence that common and rare variant of CNT NAP2 confers. So it says that CNT NAP2 confers risk to ASD or ASD related endophenotypes. So next is the role at the synapse and in autistic spectrum of disorder. It's a very, very important point. Synapse is a very, very important point. So SHANK, it's a multi-ankyrin repeat domain protein, SHANKs, which is again classified as SHANK1, SHANK2, SHANK3. So mutation in SHANK genes are potential monogenic cause for autism spectrum of disorder. So it is saying that it is the only monogenic cause, that is the monogenetic disorder, right? A single gene is enough to induce autistic spectrum of disorder. So this results in synaptic scaffolding to regulate spine morphology and neurotransmission. So neurotransmission is a very, very important factor in the autistic spectrum of disorder, because this is, I say, a child that child is abnormal, behaving abnormal, but I have never thought of that the stimulus in a child with an atypical behavior is different from a normal. And that's why they behave atypical. So that's why I am saying that SHANK gene is very, very important in the neurotransmission and in induction of the correct type of input, neuronal input, or or sensory input in the child. And from there, a lot of sensory issues may develop. So this is your DYRK1 truncation. So DYRK1 truncate mutant impairs neuronal, dendritic, and spine growth and interfere with postnatal cortical development. So DYRK1 expression profiling during brain development. The dosage of DYRK1 is crucial for the dendritic growth in cultured neurons. So it has been seen in the lab that the dendritic growth is directly dependent on DYRK1. So DYRK1 is crucial for growth of neurites, spines, hippocampal neurons in slices. So even cortical migration during early postnatal development. So when there is an early postnatal development, this particular gene has a huge role to play. So duplication of DYRK1 results in abnormal neuronal development such as compromised dendrite outgrowth and cell differentiation. DYRK1 transgenic mice has also been created. And this mouse model of DS has overexpressed DYRK1 by 1.5-fold, showing defects in learning, memory, as well as hyperactivity and altered motor learning. So proline-directed serine-threonine kinase that has been reported to phosphorylate several substrates related to gene transcription, neuronal degeneration, and synapse dysfunction. So no doubt, it is it is the gene which is responsible for so diverse spectrum of disorder. We call them diverse, neurodiversity, because the mutations, the changes in these genes are actually diverse. That's what is my conclusion. So TBR1 gene, T-box brain 1, OMIM, encodes a neuron-specific transcriptional factor of the T-box family. And it is highly expressed in the deep layers of the cortex, which involves differentiation of the subsets of projection neuron. So the importance of this transcription factor is neurodevelopment is evident from studies of mice. And if there is a mutant, there is an abnormality in cortical lamination and they die shortly after birth, though it is not that much related to autism, but homozygous mutant die, but heterozygous mutant show autistic-like behavior. Moving on to neurodevelopmental disorder associated with ADHD. So this is your LPHN3, DRD4, and DRD5. So as I have told you that SLC63 and this DRD4 VNTR is involved in G protein coupling of DRD4 neurotransmitter. It's a 7-repeat allele. It's thought to have arisen from 30,000 to 33,000 years ago from a series of rare mutational events. So Barr et al. Found no association between ADHD and 2 single nucleotide polymorphism in promoter region. This is the promoter region which is given over there, right? Observed a single overtransmission of RS1800955, an allele, and a trend towards association with RS747302C allele. So the lot of this are basically DRD4 is basically a variable number of tandem repeats. So these tandem repeats are coming generation after generation. With increasing number of tandem repeats, the their severity increases. So this is SLC6A3. It's a candidate gene for attention deficit hyperactive disorder. Again, it is having a tandem repeat of VNTR. And also, it is the polymorphism is located, change is located in 3' untranslated regions, 3' UTR. So polymorphism was identified in human coding DAT1, located in the 3' untranslated region with repeated numbers between 3 and 13, 9 and 10 repeats. Alleles are the most frequent in population. Then 5HT1B. So this is also associated 1B and 2A. See HTR1B and HTR2A. So these are the polymorphisms which have been identified, which encodes the serotonin receptor 5. So it's a serotonin receptor 5. So a total of 21 double hit SNP, single nucleotide polymorphism, passed the quality control matrices and were examined in CEPH pedigree to define the haplotype block structure in and around HTR1B locus. So HTR1B gene is both in biological and positional candidate gene for involvement in genetic etiology of ADHD. So moving on to my next question. This is a very important question. See, I have attended a lot of FDPs and there I have asked to all the geneticists that they are talking so much about genetic profiling, genetic counseling and so many things. So what about the people, this child suffering from neurodevelopmental disorder, because they need also to be established. They need also to be independent. So the answer they gave to me was your FMR1 gene test. So if you have a genetic test, which like the genetic test for Down syndrome, Turner syndrome, Edwards syndrome, if you want to have a genetic test, then you can only do a test for FMR1 gene. And this FMR1 gene is fragile X syndrome. So the X gets fragile generation after generation, and the symptoms get gets much more, severe generation after generation. So that is why, so this little screenshot is on FMR1 gene testing. So they are saying that this is basically a predictive carrier and also microarray analysis. So predictive carrier of genes which are associated with autistic spectrum of disorder. But unfortunately, it is not just fragile X. There are so many different kind of genes which are associated with the development of our brain. We have to understand the development of the brain, the development of the neuron. And whether these genes are, there is multiple mutations at a time. If it's a multiple mutation, it could not be targeted, but if it's a monogenic mutation, it can be targeted by gene therapy and antisense therapy and so many other different kinds of therapeutics. Moving on to your future of next-gen sequencing in detection of ASD. So I have also asked this question to one of the scientists, and the scientist and that particular person said that ASD, you have to extract DNA, then you have to take out their exomes, that is, you capture the exomes and do the next-gen sequencing to get the sequence, and you have to compare with your control group. And there you have to find out that which sequences are different in an ASD patient with respect to control group, to identify those sequences, and then you can treat, or you have some genetic treatment against those sequences. So that was his answer. But there are some people who are working in NIMHANS about this next-gen sequencing and genetic profiling, and we are developing a mutational landscape for ASD and ADHD patient. So my discussion and conclusion is genetic polymorphism are involved in the development of autism, ASD, and ADHD. Multiple mutation involving frame shift, deletion, addition are involved in the emergence of autism, ASD, and ADHD. FMR1 gene can be included in the genetic test for detection, but it will not give you a correct idea that, okay, this will be a child with autistic spectrum of disorder, because there is a clear difference between a Fragile X, though Fragile X is a mental retardation, but autism or autistic spectrum of disorder is not always a mental retardation cases. And future of next-gen sequencing in detection of neurodevelopmental disorder. So this is also another important point we have discussed today in my slide. So in conclusion, I have some future prospect or some questions. So these are my references. Most of them are from Nature Review, because all this work has been documented in Nature Review. I don't know why that these works are not that much encouraged by all the scientists, or they are not doing enough work in this particular field. But a time will come when we will be delving into the correct gene, their pattern of inheritance, then whether it's a monogenic or a polygenic disorder, and we will be developing some therapeutics, maybe a precision medicine, maybe a customized medicine for the patient, maybe the gene therapy, though gene therapy is not available to all stratas of the society. So these are some terms which are associated with, and also it is having a lots and lots of risks. So it is a controversial therapeutics. But if it can be a precision medicine against any kind of genes, if one gene can be pinpointed or two genes can be pinpointed, then we can delve into the mechanism of development of autism or autistic spectrum of disorder or attention deficit hyperactive disorder. That's all from my end. Thank you, everyone. So for any question, you may write it in the comment box. I will be happy to give you the answer to those questions.